Welcome everyone. Today I will be discussing the fascinating concept of the rolling circle model in genetics. This model is crucial for understanding various genetic processes, particularly those involving DNA replication and certain types of viral replication. Let us dive in. All living entities have DNA, and DNA replication occurs in all living creatures, which is a biological process that produces two identical DNA molecules. The process of replication is no wonder the foundation of genetic inheritance. Rolling circle mechanism was first proposed in 1968, also known as unidirectional replication. It is called rolling circle replication because it was first discovered in a type of phase where the template circle seems to roll. For example, the replicating DNA can be compared to a roll of toilet paper, unwindowing as it moves across the floor. The rolling circle model is a fundamental mechanism used during DNA replication as well as in certain viral replication strategies. It is characterized by the unidirectional synthesis of new DNA strands, typically forming a single standard circle or loop structure. The history of rolling circle replication dates back to the mid 20th century, when researchers first observed and described this unique mechanism of DNA replication. Let us look into some key milestone in the history of rolling circle replication. First one is discovery of bacteriophages. Bacteriophages are viruses that infect bacteria, were discovered and studied in the early 20th century. Researchers noticed that certain bacteriophages produced large amount of DNA during their replication cycle. Second one is the replicative form. In the 1950s and 1960s, scientists studying bacteriophages and other circular DNA elements observed a phenomena known as the replicative form. This indicated that the DNA was replicating via a rolling circle mechanism. Third one is discovery of 5X174. In 1962, Robert Sinsmer and his colleagues discovered the bacteriophage 5X174, which has a small single standard circular DNA genome. They noticed that 5X174 DNA replication followed a rolling circle model. Fourth one is simian virus 40, or we can say it SV40. In the late 1960s, researchers studying SV40, a virus that infect monkeys, also observed that its DNA replication involved a rolling circle mechanism. Now, see the mechanism of rolling circle replication. The rolling circle replication is completed in the 3 steps in general. First is initiation, second one is elongation, and the third one is termination. You can see the pictorial representation. The first step includes binding of a replication initiation protein called Rep-A to a section of the double standard DNA called the origin of replication or ORI. Binding of initiator protein to the 5' phosphate end of the nicked strand and the 3' hydroxyl end is the free to act as a primer for the DNA polymerase enzyme. Here you can see the pictorial representation. The second step includes beginning of the replication processes by adding DNA polymerase to the 3' end. In this pictorial representation, as DNA synthesis goes on, it can make multiple single stranded copies of the original DNA in a series called a concatemer that goes from head to tail. In the third step, you can very well see here, copies of the original DNA molecule that are made in a straight line are changed into copies that are made in a circle. Now, let us have detailed insights into the process of rolling circle. So, first one is, what is first one? First one is initiation. So, the process begins when a specific site on the circular DNA molecule is recognized by a replication initiator protein. This protein binds to the origin of replication and makes a single standard nick in the DNA molecule. Second one is unwinding. After the initiation step, the replication initiator continues to unwind the DNA in one direction, creating a single standard region called the rolling circle. In third step, synthesis of new DNA. Once the rolling circle is generated, DNA synthesis begins using the single standard region as a template. Nucleotides are added one by one to extend the newly synthesized DNA strand. Fourth one is the replication fork movement. As the synthesis proceeds along the rolling circle, a growing replication fork is formed, moving in a unidirectional manner around the circular DNA. In the next step, second strand synthesis. During the process, a complementary DNA strand is also synthesized, creating a double stranded DNA molecule with one circular strand and one linear strand. Then termination. Eventually, the replication process reaches a termination point, resulting in the generation of the multiple circular DNA molecule, each containing one original strand and one newly synthesized strand. Now, let us see the mechanism of the rolling circle model in three basic steps. The first step is the initiation, then elongation, then termination, and then again repeat. The rolling circle model is different from the more commonly known semi-conservative DNA replication observed in most cellular organisms, where both strands of the double helix are replicated simultaneously. The rolling circle model is particularly relevant in the context of plasmids and certain viruses, because it allows for the rapid amplification of genetic material and the generation of multiple copies of the circular DNA molecule. To summarize, rolling circle replication proceeds through the following steps. First one is the circular DNA will be nicked. Second one is the 3' and is elongated leading strands. Third one is the 5th dash and is displayed which lagging strand. Fourth is the replication of both unnicked and displaced ssDNA take place. Fifth one is the displayed DNA circulates and synthesis its own CS. Now, let us see the enzyme involved in DNA replication. What are the enzyme involved in it? First is helicase, which unwinds the DNA double helix. Then primase, which lays down RNA primers. Then DNA polymerase 3, main DNA synthesis enzyme. Then DNA polymerase 1, which replaces RNA primers with DNA. Ligase, which fills in the gaps. Gyrase, which help in topographical or topological transition of DNA. Now, what are the applications of rolling circle model? First one is plasmid amplification. Plasmid are circular DNA molecules found in bacteria and used extensively in genetic engineering. The rolling circle model allows for the rapid amplification of plasmid DNA, enabling the production of large quantities of the plasmid for various applications. Second one is the DNA sequencing. It has been used in certain DNA sequencing methods such as single molecule, real time, smart sequencing. Here DNA polymerase synthesizes a complementary strand in a continuous manner, incorporating labeled nucleotides. This result in generation of a long ssDNA that can be read and used for sequencing. Third one is viral replication. Many bacteriophages, viruses that infect bacteria, we all know that, use the rolling circle model for their replication. Understanding this process is essential for studying viral infections and developing strategies to combat them. Fourth one is the recombinant DNA technology. The rolling circle model is commonly employed in recombinant DNA technologies to generate large amount of specific DNA sequences. It allows for the efficient synthesis of DNA fragments that can be later used in genetic engineering and gene editing applications. It is used in molecular cloning also. In molecular biology, cloning involves the replication of a specific DNA fragment or gene of interest. The rolling circle model is often utilized to amplify and clone DNA fragments, making it an essential tool in genetic research. Next application is development of DNA probes. Rolling circle amplification is a technique used to amplify specific DNA sequences, which can then be used as probes for detecting complementary sequences in various diagnostic applications such as in situ hybridization and gene expression analysis. It is used in biosensor and biomedicine also. It can provide a unique perspective in comprehensive understanding of biological processes. It can also be useful in effective diagnosis of diseases as a drug carrier and many more. that you can see in this pictorial representation in biosensing, bioimaging and in biomedicine. In summary, the rolling circle model is a fascinating the important mechanism in genetics. It plays a vital role in DNA replication as well as in the replication of certain viruses. Understanding this model help us grasp the complexity of genetic processes and the strategies viruses employ to propagate. Thank you for joining us today. I hope you found this information enlightening.